Alright, we are starting with the description of chemoreception within smell and taste. Starting off with smell, also known as olfaction. Here's a little diagram we have here of the nose and the receptors up here. So with these little olfactory receptors, they run through the olfactory epithelium, through the bone here, the cribriform plate, and then up to the olfactory bulb, which leads back to your brain. And so the way we're able to sense smell is that we have the olfactory nerves here, and as airborne molecules come into the nose, they dissolve in a mucus, and then so first they hit the mucus and then they go and bind to these olfactory receptors, which then send signals and action potentials back through the olfactory nerve, these olfactory nerves to the olfactory bulb, which then they get sent off to the brain. As far as taste goes, we have a couple of different methods of sensing taste. There are about 5 different types of tastes that more like 5 different types of taste stimulants that we have, and we have them here. The way that they occur are different in each one, but salty and sour, which are activated by any sodium or potassium for salty, and HCl or hydrogen ions for sour. What happens here is that it's a direct bond as a ligand to the cell. And the bond will then open up channels. They'll open up depolarizing channels, so cation channels, which then go on to open up voltage gated calcium channels, which then cause the action potential to occur, or which then cause the sending off of vesicles with the neurotransmitter to then go on to the neuron. And that's for salty and sour. For sweet, bitter, and umami, which for sweet we're looking at sucrose. Sour is sweet, bitter, and umami. For bitter, it's more of a quinine is the molecule, and then umami is glutamate. And with each of those, it is a G-coupled protein receptor. And so the molecule is going to come in and bind to the outside of the cell, which then causes a cascade of signals, which goes down on to activate IB3, which then goes down and cascades to opening the smooth endoplasmic reticulum, which releases calcium and causes an increase in calcium in the cell, allowing the vesicles to leave, sending off the signal. Right now I'm going to describe a little bit about how sound waves that carry energy turn into mechanical vibrations, into fluid waves, and then chemical signals, which then send off action potentials to the brain to help us hear. With that, I'll talk a little bit about the mechanisms of hearing as well, with the inner ear and the middle ear and what their roles are. So as far as sound waves are just compressed air, or the molecules in the air. So if we have a bunch of molecules here and a wave comes by, the energy pushes these molecules, which then push further the other molecules around them, sending a wave off into the distance, further and further. And so when we get that wave into our ear canal, we have 3 different bones here. We have the malleus here, and then we have the incus here, and the stapes here. Okay, so malleus here, incus here, stapes here, and also referred to as hammer, anvil, and stirrup. So we have the purpose of the middle ear, so this section here with the bones, is to amplify the signal. So those waves come in, and they become a mechanical vibration through the eardrum, which is right here. That's what we call the eardrum, or the tympanic membrane, and the waves hit it and it creates mechanical vibrations. And when those mechanical vibrations go onto these bones here, they amplify the signal greatly. Once it's amplified, it then moves on to the cochlea. When it goes on to the cochlea, the mechanical wave then hits this little thing here called the oval window. And this whole thing is a cochlea. It comes in a couple of different parts, but when it hits the oval window, the cochlea is filled with a liquid called perilymph, which is high in sodium. And when it hits the window, it sends the vibration throughout all the fluid, and since fluid is non-compressible, it has to have a way of releasing some of that pressure. So there's the round window. It's almost like a pressure valve. So when this one beats in, this one beats out, which allows it to not cause any problems or damage within the ear when you receive sound. And so once it reaches that point, and there's a signal being sent through here, there's this thing called the basilar membrane. The basilar membrane is shaped somewhat like a xylophone here. And so just like a xylophone, the smaller the notes are, or the smaller the section of the basilar membrane is, the better it's going to resonate with higher frequencies or higher pitch. Frequency is just the amount of waves per second. And so the more amount of waves there are per second, the higher the frequency, the higher the pitch. And that resonates with the smaller section of the basilar membrane. As you go further, the larger parts resonate with a lower frequency, which is a lower pitch. And this basilar membrane wraps around half of your cochlea here. So this is it laid flat as it goes along in this picture. And starting here, it would be thin and then gets wider and wider and wider as you go further. This is important because on top of the basilar membrane, we have these things called hair cells. You have your outer hair cells and your inner hair cells, and this red line here is the basilar membrane. When the basilar membrane vibrates, if we get a closer view of this hair cell, basilar membrane vibrates, and there's this thing called the tectorial membrane up top here, and the hair cells are going to run into that tectorial membrane there, and the outer hair cells are partly connected to the tectorial membrane, but when it interacts with it, it pushes on the tallest part of the hair cell, opening up a ion channel. And since this ion channel pops open and the intersection here is filled with a lot of endolymph, which is very different from perilymph. It has a high concentration of potassium. Potassium is going to flow into the cell, depolarizing it, causing voltage Well, in this case, on the outer cells, there's these little guys that used to be voltage chlorine channels, but they kind of lost their purpose and were repurposed. These are called Preston proteins. And Preston proteins, when there's a change in voltage or it is depolarized, the Preston proteins contract and make the outer hair cells smaller. And what that does is then pull the tectorial membrane down far enough to hit this inner hair cell. And when it hits the inner hair cell is a little bit different. It doesn't have that Preston, but it does have voltage calcium channels, and it does have these ion channels on each of these little fingers on the hair cell. And when it opens, it gets pushed over, opens up, pulls each one over, opens up the ion channels. It'll depolarize the cell, allowing the voltage calcium channels to open, which then depolarizes or flow in, allowing vesicles to leave the cell. And that's going to be the signal that gets sent. So back to this picture. If you notice down here, these are your neurons that will lead off to your brain. But these ones have arrows coming in, and this one has arrows going out. So on the inner hair cells, the arrow going out signifies this is the one that's going to be sending the signal of sound to your brain. And sometimes when you're in a loud room and you want to focus in on a conversation, your brain needs to tune out noises. And so this isn't a one-time thing within your ear. This flows all the way up all the way up your cochlea, up and down your cochlea at different levels. And so there's so many hair cells throughout this entire thing sitting on the basilar membrane, and they sense for different pitches. And so when you want to focus in on a conversation, your brain is going to tell your outer hair cells through a signal to not bend as much, and that will kind of deactivate Preston, which then won't allow the tectorial membrane to interact with your inner hair cell. Therefore, it would be blocking out that sound or blocking out that signal coming in to the cell. And a little bit about the membrane here. We have this little section here. It is called the arteria vascularis, which is a portion of the membrane of the cochlea which allows the depolarization of where the endolymph is compared to the perilymph. Those sections again, we have the scala vestibuli. It's this first portion of the cochlea, separated by the helicotrema, which then leads down to the scala tympani. And then in between that section, those two sections, is called the cochlear duct in here. So the cochlear duct has endolymph. It's high in potassium, while it's inside the cochlea you have your perilymph, which is high in sodium. Coming back here, that means we need to somehow maintain that high amount of potassium, high amount of sodium in their respective sections. We do that through this little section here, and it has this special pump called the NKCC, the sodium potassium chloride channel, which pumps in sodium, potassium, and chloride to chlorides for 1 and 1 sodium and potassium, and then it does it again through the second part of the membrane, which then causes the potassium to then be recycled into the cell.